What's up, guys? How are you all? I hope you're super excited for the series where we are going to complete NCERT reading of class 11 and 12 biology as well as chemistry. Please do comment down below if you want me to do physics as well. As of now, our first target is class 11 biology. So, invite your friends, like, share, and subscribe. And now, let's not waste any more time and get started with the chapter. Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots, stems, and leaves, it is convenient to study the transverse sections of the mature zones of these organs. Dicotyledonous root. Look at figure 6.3a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hair. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water-impermeable, waxy material suberin in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis, such as pericycle, vascular bundles, and pith, constitute the stele. Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. Figure 6.3b. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem. Figure 6.4a. Covered with a thin layer of cuticle, it may bear trichomes and a few stomata. The cells arranged in multiple layers between the epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consists of round thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains, and the layer is also referred to as the starch sheet. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of dicot stem. Each vascular bundle is conjoined, open, and with endark protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. Monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath, and a large conspicuous parenchymatous ground tissue. Figure 6.4b. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. Figure 6.3 TS. A. Dicot root primary. B. Monocot root. Figure 6.4 TS of stem. A. Dicot. B. Monocot. Dorsiventral dicotyledonous leaf. The vertical section of a dorsiventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface, adaxial epidermis, and lower surface, abaxial epidermis, of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called mesophyll. Mesophyll, which possesses chloroplasts and carry out photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at figure 6.5a and find the position of xylem in the vascular bundle. Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. Figure 6.5b. In grasses, certain adaxial epidermal cells along the veins modify themselves into large empty colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles, except in main veins, as seen in vertical sections of the leaves. Figure 6.5. TS of leaf. A. Dicot. B. Monocot. Summary. Anatomically, a plant is made of different kinds of tissues. The plant tissues are broadly classified into meristematic, apical, lateral, and intercalary, and permanent, simple, and complex. Assimilation of food and its storage, transportation of water, minerals, and photosynthase, and mechanical support are the main functions of tissues. There are three types of tissue systems: epidermal, ground, and vascular. The epidermal tissue systems are made of epidermal cells, stomata, and the epidermal appendages. The ground tissue system forms the main bulk of the plant. It is divided into three zones: cortex, pericycle, and pith. The vascular tissue system is formed by the xylem and phloem. On the basis of presence of cambium, location of xylem and phloem, the vascular bundles are of different types. The vascular bundles form the conducting tissue and translocate water, minerals, and food material. Monocotyledonous and dicotyledonous plants show marked variation in their internal structures. They differ in type, number, and location of vascular bundles. The secondary growth occurs in most of the dicotyledonous roots and stems. Awesome guys, so this marks the end of the chapter. I hope you liked the video. Please share your thoughts in the comment section and don't forget to like, share and subscribe. Let's meet next Sunday to continue the series with a new chapter. Bye.